to the Institute this morning. Some of you may have been here this time last year when we ran a workshop following as part of Science on the Swan, and we've in some senses replicated that successful formula, except I've cut my own time down to 1 minute to improve the whole proceedings. We have a really nice program and a series of speakers who are all champions of their own domain, and you'll hear about some very exciting cutting-edge tools that they're using. But they're responding to unmet need in their own domains, and just a few more people coming in. Welcome. What you'll see as being accomplished using tools that range from the evolutionary to the population level, large cohorts, individual patient deep sequencing, and down to the single cell level, that these extraordinary tools, powerful tools of our time, really can be very impactful. But what I think you'll find interesting and inspirational is the creative ways in which these investigators are putting these tools together. So it's also an enormous pleasure to introduce Chris Smith, who's well known to you all, the Naked Scientist. Chris introduced me at the wonderful after-dinner talk that he gave to a term which I hadn't heard for a long time, which was pay-sake. And it reminded me that as a 15-year-old, my first holiday job was at the school, and I remember the builder saying, "Hey, pay-sake, it's cement you're making, not a bloody meringue." So it really took me back to hear that. Chris, welcome so much. It's great to have you here, and thanks for sharing the rest of the proceedings.
Thank you. Good morning, everybody. I must admit I'm a bit scienced out this week because we've had the Science on the Swan meeting, and I keep saying this, but I think that was the best sort of science conference that I've ever been to. I said it on the radio this morning, had Radio National phone me up 6:20 this morning. What have you been up to? So I gave a quick summary, and I highlighted some of the research findings. And it's interesting how many of those amazing things were just coming straight out of Australia. I shouldn't say amazing, but it is amazing, because compared with America, if you think how big the States is, and how many people there are there, compared with here in Australia, I think Australia punches very high above its own weight on the international stage. And we've just actually had the world rankings of universities announced this week, and on the front page of the BBC website this morning it says UK universities fall, in the top 100 league, and actually my own institution, Cambridge University, has plummeted 2 places. So we're still in the top 5, apparently, but we're not number 2 anymore. Harvard and MIT are pipping us to the post, but it's all much for muchness, isn't it? Anyway, I'm very delighted to be here. It's a huge honour and a privilege, and I'm looking forward to all the great talks, and I hope that you're going to be amenable to being interviewed possibly for one of our programmes, or at least tapped on the shoulder to come and contribute to one of our programmes in the future. I'm going to tell you first though, Nancy Cox is the director of the Vanderbilt Genetics Institute and the Division of Genetic Medicine, and what she's going to tell us about is her work linking genetics to phenomes. Hello Nancy.
Hello.
Over to you.
All right. So, can you guys see the screen okay?
Yeah. Yes.
Yep.
Okay. So what I'm going to talk to you today a little bit about the methods, a new method that we've been using to really integrate transcriptome and genome data with electronic health records. So that's the PrediXcan method. And then some preliminary results of the application of this method to BioVU, the biobank at Vanderbilt University. And so I'll cover briefly some novel gene phenotype discoveries, tell you more about what we are seeing as a continuum from Mendelian diseases to common diseases, and some new ways to maybe get insight into biological mechanisms of disease, as well as some really new big picture biology that I'm going to try to convince you about. So a lot of the motivation from this comes from research that we and others have done. GTEx is a big part of this, showing that a lot of the common variant contribution to common disease is coming through regulatory variants. So this is a paper from colleagues at the Broad, including somebody that used to be a student in my lab. And what they did was clever. They took 11 common diseases and just combined them together as disease, and all of the controls were combined together as controls. And what they looked for was the ability of SNPs to encode annotations to concentrate heritability. So they estimated heritability using all SNPs, and then re-estimated the heritability using just SNPs mapping to these different encode annotations. So coding, untranslated, promoter, DNase I hypersensitivity sites, intronic and intergenic regions. And you can see that about 40% of the overall heritability they estimate using all SNPs is attributable to SNPs mapping to the encode annotation of DNase hypersensitivity sites. That's just for the genotype SNPs. If you include the imputed SNPs, you get up to 80% of the heritability that you can estimate using all SNPs. You're able to estimate using just SNPs mapping to these DNase I hypersensitivity sites, and that's largely about transcriptome biology. So this maybe wouldn't have worked if this weren't such a broad phenomenon. That is, it doesn't matter that they combined all these different diseases together. This is something that is true for common diseases in general. And if it weren't, you wouldn't have seen much signal overall. I think so motivated by that, if you think about the way gene expression can be decomposed, so what we measure as gene expression through RNA-seq, there's some part of it that is just completely genetically regulated. But of course, we know that other factors contribute as well. Exposures are very important in what we measure as gene expression, and those can be very local exposures, like what you had for breakfast this morning, but also long-term exposures, like whether you're a cigarette smoker, whether you exercise regularly. And over a lifetime, the genetic regulation of gene expression and all of the exposures work together to create new phenotypes in those of us who might develop disease, for example. And disease itself, so trait shown here, can feed back on what we measure as gene expression. And we know that because when we measure gene expression in peripheral blood monocytes from kids with asthma and kids without, there are thousands of genes highly significantly differentially expressed, largely as a consequence of having asthma, not as a cause. Because of this feedback, it's particularly difficult to just measure gene expression and try to understand what genetic contributions there might be to a disease. But when we focus on just this genetically regulated part, we have basically a one-way arrow to the traits we're interested in studying and a kind of a Mendelian randomization experiment. So this is a paper that we published last year in Nature Genetics, and all of the software is publicly available. So what we've done is to take GTEx as a reference panel. So GTEx is the Genotype Tissue Expression Project. It's an NIH Common Fund initiative where up to 50 tissues have been collected from individuals, mostly organ donors. And RNA-seq is measured in each of those tissues. And there's also whole genome sequencing being done on DNA extracted from blood from these individuals to get us the DNA variation that we can use to look at the genetic component to gene expression for each of these tissues. So we have the genetic variation measured. We have the observed transcript levels for each gene in each tissue. And then we simply build linear equations. So we use elastic net, a kind of a penalized regression approach to use the DNA variants to predict the measured transcript levels. And so basically, we create a database that has for each gene in each tissue the SNPs that come into the prediction equation and the weights that are associated with those SNPs. So with that database, we can go into any data set, like BioVU, where we have just genome variation measured through, for example, either genome-wide association studies, so just genotyping arrays, or whole genome sequencing would certainly work as well. But where we can take that genome variation and the database of the prediction equation and essentially impute the transcript levels for each gene in each tissue, and then test the imputed transcript levels across all individuals with whatever traits we're interested in studying. So that's the idea of PrediXcan. We build an endophenotype that we know drives a lot of the biology of common disease, and then we use that calculated endophenotype, the transcript levels, genetically predicted transcript levels for each gene in tissues of interest, to test for association with the traits that we want to study. Now, of course, this only works as well as these expression phenotypes are heritable. So if we start with just a single tissue, this is whole blood, and we build our predictors in one data set. In fact, these were built in the Depression Gene Network data, and then test them in GTEx. We can get an out-of-sample prediction performance contrasting the directly measured transcript levels through RNA-seq against the genetically predicted transcript levels. In black, I show the heritability of all the genes that are expressed in whole blood, and in the gray bars around that, you see the 95% confidence intervals around the heritability. And when heritability is good, above 10 or 15%, you see that, so the red dots show this out-of-sample prediction performance. It's quite good. So we really see that the predicted expression is very near what you would get from the heritability expected for this particular trait. Now, in a single tissue, there's only about 4,000 to 9,000 genes that have really high quality expression, because we have a lot of genes down here with very low heritability and therefore very low predictability from the genetic perspective. But across all tissues, most genes get into this sweet spot. So we have more than 18,000 genes with a correlation greater than 0.2 in at least one tissue for the correlation between the directly measured transcript level and the genetically predicted transcript level. So as I said, in any single tissue, something around 4,000 or 5,000 genes, at best 8,000 or 9,000 genes have this kind of quality prediction, but over all tissues, most genes get into this sweet spot. So the advantages of this framework is that we're using more about what we've learned about transcriptome biology and about the fact that so much of the common variant contribution to common disease is coming through this sort of regulation. And our signal comes at the level of the gene. So rather than having SNP-based signals where we then try to figure out what the SNP is doing, our signal is at the level of the gene. We know exactly the biology it's about. It's about the genetic regulation of that gene's expression. And we have an easy to interpret direction of effect. So we know whether it's the predicted increased expression of a gene that is associated with increased risk of a disease, or the decreased predicted expression of a gene that would be associated with the increased risk of disease. And having this gene-based signal with a direction of effect really makes it much more tenable to think about real systems biology, and certainly we have a much better ability to do pathway and network analyses. This also sets up a natural framework for the unified analysis of whole genome sequence data, and Vanderbilt was just recently named as one of the centers for the analysis of all of the whole genome sequencing data being generated by the NHGRI sequencing centers in the US. So we're applying this to the big biobank at Vanderbilt University. So Vanderbilt built their own electronic health records starting more than 20 years ago. And today there's about 2.5 million subjects in what you might call a clinical data warehouse. The name that Vanderbilt uses is the synthetic derivative. It's a de-identified and continuously updated image of the electronic medical record. So there's information not just on the billing codes, but on text mining of physician notes, the pharmacy records, all of the test results that get ordered. Potentially you can get access to imaging information. So there's really a lot of data that you can use from this. Of the 2.5 million subjects in the synthetic derivative, we have DNA samples today on more than 215,000. And today we have dense GWAS level genotyping on about 20,000 and exome chip data on 36,000. But by this time next year, there'll be more than 3 million subjects in the synthetic derivative. We'll have DNA on probably more than 230,000, but certainly more than 225,000. And we'll have dense genotypes on more than 120,000 of those subjects. So more than half of our data should have genotyping, whole genome sequencing, or exome sequencing. So we will have thousands and up with sequencing as well. And having the entire medical phenome to look at allows you to do a different kind of study than GWAS. So everybody's familiar with genome-wide association studies, but what's been pioneered at Vanderbilt is what's called phenome-wide association studies, where traditionally they take an individual DNA polymorphism. It could be a loss-of-function mutation in a gene, or maybe it's a lead SNP, or a genome-wide association study on a particular phenotype, and ask for that one SNP, what associations do you see across the entire medical phenome? Associations with infectious phenotypes, neoplastic phenotypes, psychiatric or endocrine and metabolic phenotypes. So this was a SNP associated in a GWAS with Alzheimer's disease, and you see it associated with a number of other phenotypes here as well. And what we're proposing to do is take this concept of phenome-wide association studies, but think of it in the context of gene-based analysis. So the PrediXcan analysis that gives us a gene-based test, we're now applying across the phenome codes that we have. So that's about 1,600 phenome codes that we would be looking at. And you can think of this as a giant knockdown experiment where we knock down the expression of each gene in each tissue and read out the consequences of that across the medical phenome. So if we knock down this gene, what does it do? What diseases might that be associated with? And similarly, it's an upregulation experiment because we can upregulate each gene in each tissue and then read out the consequences across the medical phenome. And of course, we're not really manipulating the human beings in BioVU. We're using natural variation and the consequences of that variation on gene expression to do this experiment for us. And we think that makes BioVU a really fun and interesting in silico discovery engine. And it actually works. So if we just tally up the number of genes a person has where there are more than 3 standard deviations above or below the mean for the BioVU population that we look at. So we just tally up the number of genes a person is in the tails of the genetically predicted distribution of expression. That tally, and this I'm going to try to convince you is the only tail of a distribution you want to be in. So you want to have the least number of genes where the genetically predicted expression is in the tails as possible, because that number is significantly correlated with how many phenome codes you accumulate within the Vanderbilt University Medical Center system. So the number of phenome codes on average is 15 or so for the subjects that are in BioVU now. As you can see, there's a very heavy tail, so there are lots of people with 60, 70, 80, 90, 100 diagnostic codes over the time they're in the healthcare system, and a small number that get into the hundreds. And when you have a chronic disease, it's easy for these phenome codes to pile up. Just over the 15 years on average that we have data on someone in BioVU, they may go from being healthy to, say, having diabetes, and then to developing kidney disease, and then to developing stage 3 or 4 kidney disease, and then end-stage renal disease, and kidney failure, and may undergo dialysis, and have a kidney transplant. And each of those diagnoses generate a number of phenome codes as you go along the way. And so it's not hard to accumulate quite a number. And there are plenty of people who are still quite healthy, and certainly there are phenome codes for a normal pregnancy and a normal delivery. So there are plenty of people with very low numbers of phenome codes, and many of those may be relatively healthy phenome codes, like normal pregnancy or normal delivery. In the first 5,000 subjects that we had in BioVU, where we had dense genotyping data, we found an association with the reduced genetically predicted expression of a gene, GRIC5, with many different eye phenotypes. So you see retinal detachments, cataract, glaucoma, primary open-angle glaucoma, disorders of the vitreous body, other retinal disorders. And from the numbers here, you can see it's not the same set of individuals with just a lot of different eye diseases. This, for the most part, is completely different individuals with different eye diseases. And in less time than it took me to do the calculations on the next 15,000 subjects in BioVU, they had knocked it out in the zebrafish core, and were able to show right away that this gene is important in normal eye development. You see a little cyclops zebrafish here, and zebrafish with small, misshaped eyes. There were some other phenotypes, so you might see some hemorrhage or cardiac edema, but the predominant phenotypes are eye phenotypes. And that confirms that GRIC5 is important for normal eye development, but didn't fully explain why just reduced expression of this one gene could increase the risk for so many different eye diseases. But it turns out when they looked at the antibody to the protein product of the GRIC5 gene, it's highly expressed in the lens of the developing zebrafish eye, which probably explains the increased risk for cataracts. It's highly expressed in parts of the eye where you may develop retinal detachment. It's highly expressed in the cells that form the myelin sheath around the optic nerve. And it's highly expressed in parts of the eye that control fluid dynamics, which would account for some of the other eye diseases that are observed. So by using the PredictScan methodology across BioVU in just the first 5,000, we already have a novel gene-phenotype relationship that's been validated in a model system study in zebrafish. But there's another kind of validation that I think is even more intriguing, based in humans. So some of the people doing sequencing in Mendelian diseases are starting to describe what they see as a continuum from Mendelian diseases to more common diseases with complex inheritance, where they've identified a number of genes important in familial neuropathies. And when they sequence a new case that looks like familial neuropathies, they sometimes find an obvious mutation in one of those genes, but in the families that they don't solve, they do see a burden of rare variants across those genes of unknown significance. So milder mutations, but more of them than you would expect by chance, mapping to genes that have already been shown to contribute to monogenic forms of neuropathies. What I'm going to talk to you today about is a different kind of continuum, from loss of function mutations to deleterious amino acid polymorphisms to just reduced expression of a gene, as being another continuum that validates that this method actually works. So if we start with this gene, nuclear factor 1 X type, or NFIX, that's a transcription factor. And mutations of this transcription factor are actually associated with two different autosomal dominant diseases. So Marshall-Smith syndrome has accelerated bone formation in hands and feet, and that can lead the kids to develop fractures. They have diminished muscle tone, and as a consequence of the diminished muscle tone in their upper bodies, they often have breathing difficulties. The larynx and trachea are characterized as floppy, and they have a lot of trouble clearing fluids from the lungs, and may develop pneumonias. They have distinctive facial features, including wide-set eyes and blue sclera, and they often won't start walking until they're almost 2 years old. Speech may be absent or quite delayed, and many of them will have some intellectual impairment. The other autosomal dominant disease associated with mutations in NFIX is Soto syndrome 2. That's also associated with overgrowth in childhood, but more commonly than with curvature and scoliosis. There are also facial features associated with it. They also have muscle weakness, but they have more congenital anomalies. So you generally see some anomalies of the kidney or the heart. The heart is probably the most common. There are very characteristic eye anomalies, anomalies of the ears, including deafness. They can have some increased risk of relatively benign tumors and low-grade malignancies. A number of the children will have seizures. They'll have some intellectual disability and notable behavior problems. Speech and language disorders, particularly stuttering, has been mentioned. But they'll sometimes have ADHD and some of the insistence on sameness features similar to OCD that are present also in children with autism. If we look at just it's the breathing difficulties that often led these kids to die relatively early with these autosomal dominant disorders, but now physicians are much more attentive to the breathing difficulties and the kids are living longer. We look at the expression of NFIX in the GTEx data. You see it's highly expressed in brain. It's pretty highly expressed in different cardiac tissues and highly expressed in muscle, skeletal muscle. But you'll notice it's also highly expressed in cervix and fallopian tube and uterus, and that's going to be relevant to what I show you with respect to what you observe when we look at the reduced predicted expression of this gene across BioVU. So with predictors built in blood, in red I'm showing some of the features that are significantly associated with reduced predicted expression of NFIX that match Soto syndrome 2 or Marshall-Smith disease, Marshall-Smith syndrome. So you see the cardiac and circulatory congenital anomalies, some classic phenotypes related to the lens and congenital anomalies of the posterior segment of eye, that are very classic, especially for Soto syndrome. But you'll notice there's also congenital anomalies of the esophagus that are highly significantly associated with reduced predicted expression of this gene. That's not been reported for either of these diseases, but of course, there have been very limited numbers of children fully phenotypically characterized, and it will not be surprising if some of them actually do have congenital anomalies of the esophagus, and especially as more are characterized. It may also be interesting, so that they have these calcium, little calcium cysts in the salivary glands. That may be something that, if it is in fact associated with the Mendelian diseases, another simple thing that can be asked to sort of cement the diagnosis, because that's also very highly associated with reduced expression of this gene. But one of the things parents always ask is, okay, they're living longer now, but what's going to happen to my child? And one of the things that we might predict will happen is that the girls, as they grow into women, will start having pelvic inflammatory disease. So you see that as a strong association here. The giant cell arteritis is observed in pemphigus and pemphigoid, maybe features that as the kids get older you might see. Not sure whether these infections are a direct pleiotropic effect of the variation, or simply that infection is easier to grow in the lungs where it's harder to get rid of the fluids. Across other tissues, you see highly significant associations with facial weakness, the pneumonia, diseases of larynx and vocal cords, symptoms of respiratory system. The symbolic dysfunction is related to the particular kind of intellectual disability. You see speech and language disorders, disorders of the tympanic membrane, neural tube defects. The kidney anomalies lead to kidney disease and kidney failure even, and that's in a p-value range of 10 to the minus 5 to 10 to the minus 8, depending on the tissue. You see fractures, particularly of the ankle, foot, and patella, again ranging from 10 to the minus 5 to 10 to the minus 8, depending on the tissue. And we also see diagnoses of seizures, convulsions, or epilepsy over a range of p-values. So basically, we're able to recapitulate the Mendelian disease phenotypes in people in our healthcare system who have just reduced predicted expression. Now, most of these people don't have all of these. That you're talking about some very common things like pelvic inflammatory disease, but much rarer things like congenital anomalies being associated. But you're really seeing most, if not all, of the features of the Mendelian disease being present in people with just reduced expression of the gene. So we are creating a database of Mendelian disease genes and their associated phenotypes. This will give us kind of a data-driven model where we have a much larger range of clinical features that might be seen with these Mendelian diseases. And it will also give us some information on outcomes that might be expected as patients with Mendelian diseases live longer. But we also have hundreds of Mendelian genes that we might say are, genes that we might call Mendelian genes in waiting. So these genes are not yet characterized as Mendelian disease genes, but we see them in BioVU associated with multiple congenital anomalies, intellectual disabilities, and other really bad phenotypes. And this looks like it could be an interesting way to kind of predict what de novo phenotypes, what phenotypes might arise from de novo mutations in some of these genes that are probably generally not compatible with live birth, but will sometimes have a child live born. This could improve the ability to make diagnoses of even already established Mendelian diseases, because there'll be more phenotypes that we can say might be associated with those Mendelian diseases. But it'll also give us phenotypes that physicians can go back to the patients and see, okay, did they have those little salivary gland cysts? Because that might make it much more likely that this is due to mutations in the NFIX gene. But there's more. So here's an example of an autosomal recessive disease, acrodermatitis enteropathica, that's associated with a really nasty skin disease, basically a really blistering skin disease around all of the body's openings. In addition, the kids have chronic diarrhea, gastritis, serious behavioral problems, and anemia. It was fatal in early childhood, so most of the kids died by age 5, rarely lived past age 6 or 7, until the gene was cloned and discovered to be a zinc transporter, SLC39A4. And they tried zinc supplementation. Sure enough, within 5 days of zinc therapy, the rash clears. The chronic diarrhea and gastritis is said to clear within a week or so, and the behavioral problems are said to be greatly ameliorated within the first couple of weeks of therapy. So a nickel's worth of zinc a day turns this from an early lethal phenotype into one that is associated with perhaps normal lifespan. It's early to tell yet, but the kids who are treated do well. This gene is really highly expressed in colon, in kidney, in small intestine, in skin, in thyroid, and in brain, because this is a log 10 expression, so really, really highly expressed in some of these tissues. And with reduced predicted expression from just blood, we see the anemias, mineral deficiency. Cajun-Beck disease was known to be a mineral deficiency, and clearly zinc is implicated. Iodine hypothyroidism is known to be one of the phenotypes consistent with zinc deficiencies, and this is highly expressed in the thyroid. You see the gastritis and there's chronic diarrhea. But you also notice schizophrenia at 3.4 times 10 to the minus 9 association. So this may be how the behavioral problems observed in the children get diagnosed when they occur later in life. The cardiomyopathy, highly significantly associated. And there's a number of other phenotypes associated with so both female and male sexual maturation. So again, as to what might have been in store for these kids if they'd lived longer. If you look in other tissues, you see a whole host of skin diseases that are probably misdiagnosed. These kids, I'm sure, just had the same acrodermatitis. So the people with just reduced expression of this gene have probably the same kind of acrodermatitis, this blistering skin disease, sometimes misdiagnosed as impetigo or pilonidal cyst, pruritus, bullous dermatitis, psoriasis, so not surprising. You see type 2 diabetes, which is also a different zinc transporter, had already been implicated as a risk factor in type 2 diabetes. But we see, in addition to the chronic diarrhea and the skin phenotypes, acute renal failure, kidney disease, chronic kidney disease, kidney failure, kidney transplant, end-stage renal disease, all of them highly significantly associated as well. Also primary pulmonary hypertension, suicidal ideation or attempt, and cerebral degeneration is a known consequence of zinc deficiency. So it's really distressing to realize that there are so many people in our healthcare system who have some really bad phenotypes, significantly associated with reduced expression of this gene, when we know that zinc supplementation cures the Mendelian disease, that is very rare in the population. And how distressed would you be to develop this skin condition in say, adolescence and have to live with it the rest of your life, and the schizophrenia, knowing that a nickel's worth of zinc could have greatly ameliorated things. So this is one I think we want to get into some clinical trial work right away. So there are literally dozens of Mendelian diseases that can be treated reasonably effectively with innocuous therapies like vitamin or mineral supplementation or dietary intervention. And there will be more people with increased risk of disease due to just reduced expression of those genes than there are people in the world today who have a Mendelian disease. Because there are 10,000 times more people at risk for the sub-phenotypes that make up these Mendelian diseases when they just have reduced expression. And this acrodermatitis enteropathica is 1 in 500,000 live births, but there are more than 5,000 patients in BioVU today at high risk for the worst of these sub-phenotypes, and more than 300 that'll have multiple of the worst sub-phenotypes. So I just want to tell you about one quick other thing. So I'm going to skip through some slides.
You've got 2 minutes left, Nancy.
Yeah?
Just saying, please make it quick because you're into your last 2 minutes.
Yeah, okay, so I'm skipping through these slides, and I'm just going to go right to the opposite of disease. So that's one concept. So if we take all the diseases, all the genes associated with increased risk of myeloid leukemia, and look at whether there's something associated with the opposite, there is, it's sepsis. If we start with sepsis and look at the oppositional disease, it's actually not just acute myeloid leukemia, but all leukemias and lymphomas. And the opposite of breast cancer includes bleeding ulcers, hemorrhage from the GI tract. The opposite of bone cancer is all lung phenotypes. And so it's like the opposite of these cancers are an immune system that is really amped up, suggesting maybe that this polygenic susceptibility of the cancers that we're looking at is a failure of immune surveillance. And the other bigger picture thing is that a lot of these phenotypes are piling up on biology that we know something about. So genes and diseases end up in these axes. So kidney failure, schizophrenia, retinopathy on one end, and a number of skin diseases and Alzheimer's on the other end. And as long as you stay balanced, you're okay. But it's easy to tip over into these disease phenotypes. And even maybe a single Mendelian disease can do that. So I think by being able to look across the phenome, we see diseases piling up on these major axes, raising the possibility that we could try to do biomarker development to keep people healthy. If we could develop some drugs that keep people from falling off of these axes. So this was just results on the first 20,000 people in BioVU, about 18,000 after QC, and we'll have more, and up to more than 120,000 next year this time. So all my colleagues and the zebrafish people and the BioVU people and our GTEx team and the GTEx project. Thanks.
Thank you very much. Quick question from me before we ask this audience. So how do you distinguish between cause and effect? Because if you see genes changing, how do you know that they're causing the phenotype, rather than if you have a horrible rash and your skin goes like that child's skin, you may have lots of gene changes that you detect subsequently secondary to the rash.
Sure. So if we were actually measuring gene expression, that would be a concern, but we're not. So we measured gene expression in the GTEx data, and then we used GTEx to build SNP-based predictors of gene expression. And it's only that genetically predicted expression that we're looking at here. So it is a one-way arrow, because it's only data based on genome variation, not measured transcript levels in these people. So this is their genetic propensity to having reduced expression of a Mendelian disease gene that we see associated with the phenotypes that make up that Mendelian disease.
Questions from the room? You
Nancy.
have the microphone, Simon, so then Nancy can hear it. We're just getting a microphone to the person so you can hear it, Nancy.
Sorry, Nancy, I'm Hugh Dawkins. I'm from the Department of Health in Western Australia. Can you expand a little bit on the work that you're doing with the Undiagnosed Diseases Network that Vanderbilt is running, and how this database is so critical for activities like that?
Yes, so with John Phillips and his colleagues that run the Undiagnosed Diseases program at Vanderbilt, we're trying to use this to prioritize the genes that come up in the sequencing studies by looking at, so a lot of times if you're looking at a single individual and you do a sequencing study, there are 12, 18 genes that are all candidates for having caused that disease. You don't want to make mouse knockouts for all of those or zebrafish knockouts for all of those. And so we're using this information, we're able to prioritize the genes that have phenotypes in BioVU that are similar to those observed in the patient. So we started out, so John was very interested in doing this, and we started out working with them, but then when we were funded to do analysis for the NHGRI sequencing centers, we'll also be working with the Mendelian disease groups that are funded through the NHGRI sequencing centers to allow them to get a better idea of all of the phenotypes that might be associated with any particular Mendelian disease, and then also to know about these Mendelian diseases in waiting that might help solve some of these a little bit faster.
Who's next? Simon.
Thanks, Nancy, and I'm sorry we had to race you along. You've got so much data pouring out of this resource that it's hard to do justice to it all. Hopefully you'll come back next year and update us and tell us a little bit more. This concept of the opposite of disease is really fascinating and really quite independent to the genetic analyses you could do. There's a lot of scope.
It's about it's about the phenome. Being able to look across the entire phenome completely changes the kinds of inferences that you can make. You'd never be able to discover that if you just studied a single disease. You can't discover it when you study just a single disease. So that's the beauty of the phenome.
Thanks, Nancy. Nice talk. I'm Christian Pfluger at the University of Western Australia. I have a question. Thinking about like big picture, long term, many more data in your database. Looking at understudied or not even really characterized genes, the more SNPs and variations you pick up, I wonder if you could then even predict sort of a gradient of what's bad and even worse of a SNP and what it could possibly mean for domain functions, etc. in these kind of genes. And maybe you can comment on that, if that is already possible with something that's already known and sort of testing against it.
So one of the things we can do is try to use more information from something like ENCODE as we build the prediction equations. Right now, those are just done statistically, and we maybe can use more information in trying to improve the quality of the predictions. I think one of the really interesting questions on the continuum between Mendelian and common disease is how often there may be people whose manifestations seem particularly severe, or somebody just having reduced predicted expression of a gene that might also actually be carrying a rare coding variant of that gene as well. And so I think as we have more people who are sequenced in BioVU, it will be possible to get a much better understanding of how the common regulatory variation may interact with the coding variation to alter the severity of phenotypes, for example.
Can I ask a related point, which is if you've got a particular SNP pointing to a disease, but then you happen to have some kind of compensatory mutation elsewhere in your genome, which balances things out, would your present system pick that up? Would you know?
So that's going to be one of the really fun and interesting things to try to find out. So I showed the slide where people were balanced, okay? But we can start really looking at that. So are the people that we can say look more balanced to us really not going into disease? You know, can we find enough genes on some of these axes that we can really start to look at that and make accurate predictions even about things like age at onset? So it'll take more data, but maybe next year this time.
We look forward to it. Any other questions? We've got time for probably one more. Shoot.
Thanks, Nancy. Just a very quick one. I'm Monica Chopner here from IIID. If you analyzed your data, did you have a specific look at the differences between the males and the females in your Phenome database? Well,
so yes, we've been actually looking at this in GTEx for a couple of years now. It looks like most of the sex differentiation in gene expression is more or less invariant, not variable across individuals. We find surprisingly few sex-biased EQTLs, for example, and most of the ones that are being observed are on the X chromosome. So it wasn't necessarily what we expected, but it's not necessarily surprising given the way the biology works out. So for the most part, the sex bias in disease may be more about invariantly differentially expressed genes between males and females than ones that have a lot of genetic variability in how differentially they're regulated between males and females.
Nancy, thank you very much. It's been a pleasure.
All right, thank you.
Now, the next person...
So I just sign off here now?
The next person we're going to hear from is Justin Balco. Now, he's actually pre-recorded his presentation for you, but he's available then to take questions live, obviously, afterwards. So this is Justin Balco, also from Vanderbilt.
Give me a minute.
In a minute.
So one of the things I might mention is it's been so energizing having Nancy come to Vanderbilt. As you can see, the number of questions and opportunities that are pouring out of this resource exceed the ability of any of us to interrogate. And so if those of you who would like to be connected to this and to have questions that you might like to get involved in, you should try and exploit the very strong collaborative relationship we have between Perth and Vanderbilt.